Hello, this is Chris again. We're going to talk about what sort of phenotype information you can get from a lactose fermentation tube here. So this is the starting culture that you would inoculate with your bacteria of choice. You should notice there's an upside down glass tube in the middle. That's called a Durham tube. And that's purpose is to collect any gas that may be formed during metabolism of the media in this case. We have lactose in this particular media, but you can have many other different types of sugars present and determine whether or not your bacteria can metabolize that sugar. Other contents that you'll see inside this media is that you have a reddish tinge to it, and that's because inside the media is a pH indicator, phenol red. And it'll be an indication of whether when the fermentation of the sugar that you also have in here results in a change in the pH of the media, you'll see a color change. And what we have are some examples of the color change that you might see from the starting red color. If you produce acid during the digestion of your sugar, in the case of lactose, you can get the formation of protons that will change the pH indicator, the phenol red, to a yellow color. If, on the other hand, you produce a base, you can get a color change that will become more of a hot pink or fuchsia colored relative to your starting red color. So that's an indication of a basic pH change compared to your acidic pH change, which would be yellow. The other information you can get from incubating your bacteria in this media is whether or not you can produce any gases. And in this case, we have E. coli that's been incubated into this the media, and the result is, first of all, you see the yellow color in the media, so you say, oh, it's taken the lactose and produced acid from that fermentation. And you also notice that inside the Durham tube, there's been an accumulation of gas, and so you know that gas has been produced in the fermentation of lactose, typically CO2 or hydrogen gas. So that's an indication of how that particular bacteria will ferment the sugar that's present in your starting material. In this case, we also have, besides the production of base, we also have a little bit of gas formation there. So that might be of interest to what observations you write down about that particular bacteria.